Hello. We're going to begin Lecture 3 by expanding upon the chemical properties that we've discussed in our previous lectures. I'm going to ask you to apply your knowledge of bonding types to understand the chemical properties of biologically important functional groups, to identify macromolecule types and specific monomers by labeling the functional groups on those molecules, to name and describe the functions of those macromolecules, and then describe the forces and bonds that provide macromolecule shape and the importance of that shape to function. So the best way for us to begin is with a review of the chemistry that you already know. There are 14 questions posted on Socrative that walk you through a lot of the most important chemistry that we've already discussed. Remember, we're going to build on this material. So the things that we did in a previous class, if you didn't understand them, that's going to make it harder for you to understand the next lecture in the series. So we want to demonstrate this for you and also give you an opportunity to better understand these chemical foundations which are going to pervade the rest of the semester. So pause the video here and do the next 14 questions. Try doing them without your notes available. See if you really do understand what's going on. And remember, this is the last time I'll remind you, but it's extremely important to know you are not punished for getting these questions wrong. What it should do, though, is highlight for you the importance of the fact you need to go back and review that material again. So pause the video and try the next 14 questions. Resume it when you're done. Okay. So hopefully by now you understand that covalent bonds are going to be the strongest and the most stable type of bond. Although it's also going to be important for us to understand that covalent bonds don't occur in a vacuum, that there can be hydrogen bonds that are also available. And when you get a large enough number of hydrogen bonds, they can also provide a lot of strength. And we're going to see that today. Now, which of the following is the best to describe electronegativity? It's an atom's affinity for electrons. And a polar covalent bond occurs when atoms on either side of a covalent bond have different electronegativities. This is because the more electronegative atom is going to have greater affinity for electrons, and that electron is going to spend more time around that more highly electronegative atom. That institutes a partial negative charge because the electron brings that negative charge with it. And this would be oxygen and nitrogen when we're dealing with biologically important molecules. Now, a molecule that is composed of polar covalent bonds would be able to interact with neighboring charged molecules via hydrogen bonds. So these hydrogen bonds can be via partial charges, which is most common, or the partially charged hydrogen, which is positive because it's weakly electronegative, can interact with, let's say, an ion in this solution. What kind of bond forms between hydrogen and oxygen? This is a polar covalent bond. Just because hydrogen is involved does not mean that there's a hydrogen bond. Hydrogen bonds are a very specific type of magnetic interaction. It's partial positives and partial negatives that are coming into contact with each other. And it's that like positive and negative opposites attract kind of thing. It's not an actual sharing of electrons. When electrons are shared, such as a hydrogen-oxygen bond, that's a polar covalent bond. Now, which of the following most accurately represents the bond? You need to show those partial charges. Remember, oxygen is more highly electronegative, so it gets the partial negative charge. When we look at a similar carbon-hydrogen bond, we're still sharing electrons, so it's still covalent, but since both atoms are weakly electronegative, this is a non-polar covalent bond. And so to represent a non-polar covalent bond, we do not use partial charges. There is no partial charge here because the electron is equally shared across the bond. Now, a molecule that's composed of carbon and hydrogen bonds is going to be composed entirely of non-polar covalent bonds. That means there's no partial charges. No partial charges mean that there's no partial charge to interact with that water, which means that this is false. There's no hydrogen bonding going on. So how do we name that? that would be a molecule that would be hydrophobic. Entirely carbon and hydrogen containing molecules we're going to learn today are what are known as hydrocarbons, and they're unique for that hydrophobicity. If you take a look at the molecules that are provided here, what you'll notice is that molecule A is an example of those hydrocarbons. It's composed entirely of carbons and hydrogens, or carbon-to-carbon bonds. All of these are going to be nonpolar, which means no partial charges, so this is not capable of hydrogen bonding with water. Sodium chloride is going to be an ionic compound, which means that these will dissociate into an anion and a cation. These are going to be charged, which means they're capable of interacting with the partial charges of water. Carbon to oxygen is going to be an example of a molecule that, even though the two sides of the molecule are the same, the fact that the bonds are not occurring from oxygen to oxygen, but oxygen to carbon, means that each one of those bonds is a polar covalent bond, with the oxygens having partial negatives and thus capable of hydrogen bonding with water. D is another example of a hydrocarbon, still hydrophobic, still not hydrogen bonding with water. And E is another example of a molecule with multiple polar covalent bonds. Nitrogen to hydrogen, carbon to oxygen, these are all going to be polar covalent bonds. There's going to be partial charges that are formed in this molecule, and so this is an example of a hydrophilic molecule. Now, I asked you to give a short answer where you chose one of these molecules that can hydrogen bond to water, and then place that molecule into a beaker surrounded by water, and that molecule would seemingly disappear. When I grade you on short answer questions that are dealing with water, I want to be very clear about my grading criteria. The first is that you need to use the word hydrogen bonds. And you need to use the word hydrogen bonds in a context that accurately describes hydrogen bonding. When you do so, you need to make sure that you're using vocabulary words. Your first set would be to correctly identify the property. In this particular situation, it would be describing water's versatility as a solvent, because water is a versatile solvent. It's capable of hydrogen bonding with many different compounds. Now, that hydrogen bonding is going to form a shell around the outside of that molecule known as a hydration shell. And that's why the molecule seems to disappear, because it gets absorbed into the matrix of all those water molecules instead of aggregating together into a visible chunk of that powder. So today we're going to start with a discussion of carbon, and then we're going to look at how we can attach functional groups composed of other atoms onto that carbon backbone, which is really how we create the diversity of organic molecules necessary for life. We'll look at reactions known as dehydration synthesis and hydrolysis that form and break apart some of these individual macromolecules. And then there are going to be 2 videos that walk you through the 4 important classes of biologically important macromolecules, and really taking a deeper dive into their functions, how they're formed, what kinds of enzymes are responsible for that. But first, we have to have a better understanding of carbon, and the fact that carbon and the ability of carbon to form the number of different molecules that it can, the diversity of bonding that it can, is analogous to the importance of hydrogen bonding, where hydrogen bonding describes why water is the ideal medium for life on Earth because of the properties that hydrogen bonding gives to water. Well, carbon and the properties that carbon brings to the formation of larger molecules called organic molecules really helps to describe the backbone for which all living things are formed from. So you really need to have an understanding of what this carbon-containing compound is, what these organic compounds are like, in order to be able to start to assemble larger living things. When you take a look at a cell, all it really is a carbon-based sac of water. Water describes how all of the things exist and interact with one another within the cell. And all of those interactions are going to be occurring between carbon-based things. Now, carbon is going to be really interesting because carbon has the ability to form a lot of really diverse molecules. And that diversity provides a nearly unlimited number of shapes. And those shapes are what actually provide structure and function. So it's easy to think about cute little cells talking to one another and actually having, like a conversation, right? Your brain tells your knee to grow bigger when you're going through puberty, but it doesn't actually work that way. What happens is a cell produces a hormone which travels through your body and actually physically touches another cell. And it's going to be the shape of that hormone that determines who it can touch and what that final interaction is going to be. Shape, the shape of a particular molecule is its function. Now, carbon is unparalleled in its ability to form these diverse compounds. That's why all living things are carbon-based. Carbon is going to have 4 valence electrons, meaning the number of electrons in its outer orbital. And the fact that it wants to have 8 means that it can form 4 different bonds. But those 4 bonds can be with many different atoms. In this class, because we're dealing with living things in cells, you're primarily going to see bonds that are carbon to carbon, or that also include hydrogen, oxygen, and nitrogen. And remember, these are going to be the 4 molecules that we've been talking about and describing in terms of electronegativity, which is nice for you. When you consider the fact that it can form these 4 bonds, you can also take a look at the shapes and arrangements of these different molecules. Not only can we consider 1 versus 2 versus 3 different carbons, but you can start to look at carbons that are going to be branching formations. You can look at carbons that include double bonds, carbon to carbon, or even that form into rings. There's a lot of diversity in how we can actually arrange them. And even though it doesn't come into play, you can't really visualize that from a 2-dimensional shape. When you start to bring this in to a 3-dimensional shape, what you can see is that we start to get structures. Now, this image on the right is showing you a much broader, much bigger form, but you can start to see how each one of these little individual molecules creates different shapes. And each one of those different shapes is kind of fitting into a different part of this cellular matrix, and they have a different function that's a part of them. So here's my question for you. What is the importance of a molecule's shape? Tell me in Socrative, and pause if you need more time. Yes, this is function, okay? Shape gives us function. Don't forget that, okay? It's not gonna go away. We're gonna keep using that. But now what I wanna do is expand upon this. Not only does carbon provide structure, and that structure gives us function, but we can attach things onto that carbon backbone made of very specific groupings of atoms that provide additional properties. So we're going to take a look at 6 of these different functional groups very quickly. You should always be asking yourself, is this functional group polar or nonpolar, hydrophilic or hydrophobic? And is there any special property that it provides? Now, in your textbook, there we're also going to include methyl groups or CH3 groups as a functional group, but they don't provide a function. CH3 or just plain hydrogen are inert. They don't participate in chemical reactions. They don't participate in bond formation. And so we don't include these as a functional group because they're not really serving a function. We're looking only at groups that provide functions. So here's our first one. This is a hydroxyl group. You might recognize that this sounds very similar to a hydroxide, which is an OH anion. But this is going to be an OH, an oxygen and a hydrogen attached to the molecule. It's polar and hydrophilic. We know it's polar because oxygen to hydrogen is going to be a polar covalent bond. We're going to have partial charges that form, which creates a hydrophilic molecule. Then we have carbonyl, a carbon to oxygen double bond. And this can be interior in the molecule or at the end. Regardless, it's always going to have a polar bond and be hydrophilic. But when you take a hydroxyl and carbonyl group and place them together, you can have something that's known as a carboxyl group. Not only is a carboxyl group going to be hydrophilic because it's composed of multiple polar covalent bonds, but it's also going to be acidic because that terminal hydrogen is going to be willing to be donated to form hydronium ions, which releases free hydrogens and produces an acidic solution. On the flip side is going to be an amino group, an NH2 group. Because this group is willing to accept a hydrogen from water, that leaves a hydroxide ion behind, and it produces a basic solution in the water. Sulfhydryl groups are composed of a sulfur and a hydrogen. This is going to be the only functional group which is hydrophobic, because hydrogen to sulfide is a non-polar covalent bond. We have no partial charges. These are chemically reactive though, and that's due to the nature of sulfur. So it's a little bit different than, let's say, the non-polar nature of a methyl group. But we're only going to see one particular type of reaction, at least in this class, and that's through cross-linking. So multiple of these sulfhydryl groups can connect with each other, and they can actually form linkages in protein structure. An example of this would be the fact that the reason why some people have curly hair is because they form these sulfhydryl cross-linkings in the proteins that are found in their hair. And lastly, the phosphate groups. So phosphate groups are going to be composed of a phosphorus molecule surrounded by 4 oxygens and 5 bonds. So one of those bonds is going to be a double bond, one of those interactions with oxygen. And then the other 2 oxygens are going to have a negative charge. Oxygen would like to have two bonds to share 2 additional electrons. And because the fact that in this instance it has a single bond, then we would assign it a negative charge. So feel free to invite you now to pause the video and check your understanding. What is the difference between an organic phosphate and an inorganic phosphate? And see if you can check yourself to draw an inorganic phosphate in your molecule, in your notebook. So does it look a little something like this? Remember that organic actually tells us that the functional group is attached to a carbon. So in this case, you would remove that carbon from the molecule, you would keep the phosphorus and 4 oxygens, you would keep the 5 bonds, but all 3 of the oxygens with a single bond would get a negative charge. Now, this is going to come into play when we start to talk about adenosine triphosphate. Adenosine triphosphate is an organic phosphate molecule. It's composed of 3 phosphate groups, hence triphosphate. And when you pack all of these phosphates together into a single molecule, what really happens is you bring energy, lots and lots of energy into this molecule. That is really the defining feature of phosphates. They provide energy containing compounds to a molecule. And this is due to the fact they have a very high free energy. And we are going to talk a lot more about this when we get into metabolism. Now, the last thing I want to do is introduce you to the way in which we form larger macromolecules. And that is through the process of combining single building blocks called monomers, some of which have their own functions, into larger compounds called polymers, composed of many of these different building blocks. When you compose these all together, they get new functions. And so these really become what we think of as biologically important macromolecules. Macro means big. Now, in order to synthesize these 2 together, we're going to use a reaction known as dehydration synthesis. D to remove and hydration referring to water. So an enzyme is going to catalyze this reaction, and it's going to be removing 2 hydrogens and 1 oxygen. Remember, this is actually going to be the molecular compound for water, right? H2O. So we're removing 2 hydrogens and 1 oxygen. Now there is a little bit of a misinterpretation here, and that is that this particular image is showing you some terminal hydrogen just floating out there that we can remove. But in actuality, the only hydrogens and oxygens we can remove are going to be coming from functional groups. That's one of the important aspects of these functional groups, is that they're chemically reactive. So if you were to go through and to try and identify and draw this interaction, you would need to start by identifying the functional groups in your monomers. And once you've identified that, you would subtract 2 hydrogens and an oxygen and then draw a new bond between the remaining components. The reverse of this process, breaking a polymer down into individual monomers, is known as hydrolysis. Again, hydro like water, lysis means to break down. So it's the addition of water which breaks the bond. Again, this is going to be catalyzed by an enzyme. All of these reactions are. So when you look at hydrolysis, you can see, for example, that when water is introduced back into the system, we're doing the reverse of that dehydration synthesis. Now, here's a review for you of all of the things that we've done, and here's my recommendation for what you should be doing. There are 8 Socrative questions that are available to you next in line that allow you to test your knowledge of functional groups. Then I want you to watch the Amoeba Sisters video that comes right after this one to introduce you to macromolecules. It's a very nice visual way with cute little cells that's going to talk you through the importance of these different groups. And then finish reading the macromolecule chapter and taking notes now that you have a base understanding if you haven't done so. There are 2 additional videos that will be posted after this that are a rapid review of each one of those classes of macromolecules. But I want to remind you that we're going to start to assemble these macromolecules into cellular components. So you really need to pay attention to the enzymes that are synthesizing the polymers and the functions that both monomers and polymers form based on their structure. So you have to understand their structure to get at their function, which means you're going to have to go back to functional groups, back to the enzymes putting these things together, and back to the types of bonds that are holding them together to understand their properties. And a good way to succeed in this class is always trying to apply the lecture to the lectures that have come before it. All right, see you for 2 more videos.